therefore gets pushed further away. Now, if that happened, then the cancer cells would die. But they actually have the ability to be able to attract the growth of capillaries into the tumour mass. So as they get bigger, more and more capillaries grow into it, supplying them with the oxygen that they need. Then the last one is evading death, very similar to the immortal one, only this time I've done a blank. Joe, help me, please.
Which one?
Evading death. Apoptosis, that's right. All cells are programmed to die after a certain point in time, and this is called apoptosis. And again, it's a signal that triggers enzymes to break down the cell membrane, and then they can get into the cell, and they basically destroy the cell. And the contents of the cell become engulfed by the cells around it. Cancer cells, those triggers are ignored by the cancer cell. They simply don't happen. So those characteristics are important because that's what makes cancer so successful inside our body. And a lot of the research that's going on around the world is looking at those characteristics and seeing what can we actually do to interfere with them or reverse them so that the cell, rather than growing forever, actually dies the way it's supposed to. And you're going to hear some of those things in Joe's and Peter's presentations today. Okay, so to sum it all up. There's a normal body cell. Mutation in it means it's starting the changes that are going to lead to cancer progression. First thing, uncontrolled growth, forming a mass of cells called a tumour. The tumour needs blood supply, so capillaries grow in and through it to make sure it gets the oxygen and nutrients it needs. And then cells break off from that tumour and migrate to other parts of the body and form secondary tumours. So that's cancer progression, and Joe's going to come back to that a little bit later when she talks about her work. So now I'm going to hand over to Joe, who's going to introduce you to the work of the Breast Cancer Research Group at LENScience.
Thank you. Hi, I am going to tell you a little bit about some of the work we're conducting here at the LENScience Institute and the Breast Cancer Research Group, where the main focus of our research has really been identification of novel therapeutic targets for treating breast cancer. And I'm also going to tell you about some of the technologies that we've been using to develop these therapeutics. Hopefully this is going to link in with some of your curriculum. I should just point out that some of these therapeutic targets that we're looking at are going to be useful in a wide range of cancers, not just breast cancer. So we're also looking at lung, prostate, and colon cancer. However, breast cancer has been the focus of our research, and that's what we're going to concentrate on today. Now, breast cancer is now the most common cancer or female-related cancer in New Zealand. 1 in 9 New Zealand women will suffer from this disease by the time they reach the age of 85. That's a very high statistic. And the incidence of breast cancer worldwide has actually been increasing. So over the last 50 years, we've been seeing an increase in the incidence rate around the world. There is some good news, however. Over recent years, we've also been seeing a decline in the incidence of breast cancer in some countries. And this is probably due to identification of risk factors such as hormone replacement therapy, which was used to treat some of the symptoms of the menopause. And that's been identified as a risk factor, and that's reduced some of the incidence. We're also actually seeing a decrease in deaths arising from breast cancer. And this is due to better detection, improved diagnosis, and earlier diagnosis and improved therapies for breast cancer. Breast cancer in New Zealand is still a major cause of cancer-related deaths in women, though. I've put up here a world map showing the rates of incidence or breast cancer incidence around the world. And when you actually look at this map, you can see some very interesting trends. So breast cancer has been associated traditionally with Western countries. So you can see increased rates in North and South America, in Europe and Australia and New Zealand. And in fact, the United States has the highest rate of breast cancer in the world, and New Zealand is in the top 5. Now, traditionally, Asian and African countries have had lower rates of incidence of breast cancer. Recently, though, we've also been seeing an increase in the rates of breast cancer in these countries as well, or some of the Asian countries. Now, the reasons for these regional differences and for this increase in some of the Asian incidents, or numbers of incidents in some of the Asian countries, is not really clear. There are a number of risk factors which go part way to explaining this, but it's only part of the story, and there are a lot of risk factors which haven't been identified yet. Very shortly I'll go into some of the risk factors for breast cancer and we can go through that. This regional difference is not down to genetics, and it's thought to be due to westernisation of environment, diet and cultural practices. Now, before we go on, I'd just like to introduce to you the hormone estrogen. So as this will crop up in some of the slides coming up, so I just wanted to reinforce this idea to you. Estrogen is, of course, the female sex hormone, and it's essential for primary and secondary sexual characteristics developed in women. It's also essential for normal breast development, but estrogen is also a major risk factor for breast cancer. So it actually promotes proliferation and survival of breast cancer cells. And that lifetime exposure to estrogen is thought to be a risk factor for breast cancer. But what are the other risk factors for breast cancer? What do we know about them, and why have we been seeing an increase in incidence around the world? Well, the fact of the matter is that we really don't know the answer to that question. There are a number of established risk factors which have been identified for breast cancer, and I'm just about to go into some of those. I'd just like to point out, of course, that risk factor means it increases your chance of getting a particular disease. Just because you have one or more of these risk factors doesn't mean that you will definitely get breast cancer. Now, as I said, there's established risk factors which have a lot of research behind them. But when we take all of these into account, they only account for 1/3 of cases of breast cancer. So clearly there are a lot of risk factors for breast cancer that we haven't identified yet. And that's in the subject of ongoing research. So let's have a look at some of these risk factors for breast cancer. So the first is being female, and that might seem obvious, but men can get breast cancer as well. It's just much more rare. And some of the risk factors I'm going to discuss are actually appropriate for male breast cancer, or are also involved in male breast cancer as well. You're more likely to get breast cancer as you get older, and the majority of cases, or 80% of cases, actually occur in women over the age of 50. It is possible to get breast cancer at a much earlier age though, and you should be aware of that. A small proportion of cases are associated with genetic factors or a family risk, family history. 5 to 10% of these are associated with mutations in genes such as the breast cancer susceptibility genes BRCA1 and 2. However, most breast cancer cases are sporadic and have no known associated risk factor or genetic risk factor. There are reproductive factors which are associated with breast cancer risk. So as Deb mentioned in the first seminar, we're going through puberty earlier and menopause later. And this lengthens or increases our lifetime exposure to estrogen. And estrogen, is, as I said before, is thought to be a risk factor for breast cancer. We're having less children, and we're having them at a later age. Having more children, or having your first child below the age of 30, is actually protective for breast cancer. And as I mentioned earlier on, other factors associated with estrogen, such as HRT, so hormone replacement therapy, which includes estrogen, has actually been identified as a risk factor for breast cancer. Alcohol is a significant risk factor for a wide range of cancers, including breast cancer. So as little as 1 or 2 drinks a day can significantly increase your risk of getting breast cancer. There are birth and growth parameters which are associated with cancer risk. So being born with a high birth weight and a high birth length slightly increases your risk of getting cancer later on in life. Now, this is actually a really interesting point, and because this points to conditions during pregnancy which have impacted on your chance of getting breast cancer later on in life. And if you remember back to the first seminar where Dr. Deb Sabloda was discussing the in utero environment and maternal nutrition and how that can impact on a number of different diseases, including diabetes, obesity, and cancer. Being taller also slightly increases your risk of getting cancer, and that's a wide range of cancers, again, not just breast cancer. And you can see here, if we plot the average height of a country against its breast cancer incidence, that we see a fairly good correlation with the tallest countries having the highest rate of breast cancer incidence. And here's New Zealand. Breast density. This is actually a significant risk factor for breast cancer, and it's something that is picked up by mammography, so by a scan. Breast density really refers to the glandular component of the breast, so the epithelial and stromal component compared to the adipose or fat tissue, and a higher breast density increases your risk of getting breast cancer. Weight gain and obesity, particularly weight gain post-menopause, is a risk factor for breast cancer, as well as many other cancers. So those are some of the established risk factors. And as I mentioned earlier on, when we take them all into account, they really only account for a third of cases. I'd just like to point out that this isn't a comprehensive list of established risk factors, and there are links on the wiki site which will direct you to breast cancer sites which can give you more information about these risk factors if you're interested. So they only account for a third of cases, and there are some possible risk factors or suspected risk factors which are currently under investigation. And I'm going to touch on a few of them really quickly, not in detail. If you want to, you can ask me about them afterwards. I'd just like to reinforce the idea that these are not established, and actually a couple of them are quite controversial. So the first is stress. Stress is thought to increase your risk of getting cancer. It's very, very difficult to dissect out all the individual components of stress, though. So although research has suggested this, it hasn't been established as a risk factor. Exposure to light at night. Right, this is an interesting one. Countries are becoming more industrialised, and that also correlates with breast cancer incidence. Now, artificial light at night actually disrupts your circadian cycle, so that's your body clock. And disruption of your body clock has been associated with a wide range of diseases, not just breast cancer. So that's actually another interesting one. Now, the last one is environmental contaminants, and this is very controversial. There's a huge debate going on overseas as to how much some of these contribute to breast cancer. And it's much more prevalent in the media over there than it seems to be in New Zealand. So some of these environmental contaminants are endocrine disruptors. And endocrine disruptors mimic the actions of hormones or disrupt hormone action. Some of these endocrine disruptors, these chemicals act like estrogen. So you can see why quite a few scientists are worried about them. But again, highly controversial. There's not really any good evidence to show that compounds like bisphenol A contribute to cancer in humans. Now, a lot of those risk factors I discussed earlier on, there's nothing really you can do anything about them, but some of them are modifiable. And in fact, it's thought that a large proportion of cancer is down to modifiable risk factors. So what can you actually do to reduce your risk? And these factors count for a wide range of cancers and diseases, not just breast cancer. So the first one is a healthy diet. Although there hasn't really been any good evidence to suggest that any one group, food group is involved in breast cancer risk or increased or decreased breast cancer risk, a Western diet, so a high-fat Western diet has been associated with breast cancer. So a healthy, balanced diet can reduce your incidence rate or your risk rate. Exercise, as I mentioned before, obesity, weight gain, of course, they are risk factors for breast cancer. So exercise and keeping a healthy weight will reduce your risk of getting breast cancer. And regular screening and early detection is also important. Again, I will put links on the wiki site, 2 sites which can give you more information about this. Now I'd like to pass on to Peter Lobe, Professor Peter Lobe, who heads up our group, and he's going to tell you a little bit about therapeutic strategies to treat breast cancer.
Hello. So a few hundred years ago, the Italian Renaissance painter Raffaello painted this portrait of his lover, and he reproduced her in quite exquisite detail. If you see here, you can see some dimpling of the skin, which is known as orange peel skin, which indicated that this lady actually had breast cancer. Now, Raffaello didn't spend 10 years at medical school and specializing perhaps like some of you kids will once you graduate from high school. He was an artist, so he probably didn't even know that she had breast cancer. But even if he did, in those days, there was probably very little he could do about it. However, modern medicine has dramatically changed the outcome for women with breast cancer. Since the time of Raffaello, relatively sophisticated surgical techniques have been developed which allow primary tumours to be removed with ease, and women are often given combinations of radiotherapy or chemotherapy to help prevent the return of disease or to kill off any residual disease that the surgery hasn't removed. Also, in recent times, we've seen the advent of so-called targeted therapies, which is really what Joe will be talking about next. That is, therapies directed against a specific molecule involved in driving a cancer. Chemotherapy has been used to target rapidly dividing cells, but it does so in a relatively non-specific way. So it also targets organs where cells are rapidly proliferating, which leads to the side effects of chemotherapy. And some of the more, at least visual side effects, perhaps not the serious ones, but the more visual side effects, such as hair loss because skin is a rapidly dividing organ, and vomiting due to gastrointestinal erosion, since your gastrointestinal tract is also a rapidly dividing organ. Given that, though, chemotherapeutics have been relatively successful in killing off rapidly proliferating tumour cells. Targeted therapies, as I just mentioned, though, are a little different in that they target a specific molecule which is involved in driving cancer progression, as we heard before, this link to form the tumour mass, angiogenesis and metastasis. They are potentially more effective as by targeting a particular molecule involved in cancer, they can actually inhibit more than proliferation. Maybe perhaps, for example, they inhibit some of the other characteristics of cancer like Michelle explained to you. And in particular, targeted therapeutics that can inhibit cell survival are particularly effective. Targeted therapeutics are also potentially less harmful to cells as the targets that we would be interested in are potentially increased in expression on cancer cells compared to normal cells, or the genes encoding these particular targets are amplified in cancer cells as opposed to normal cells, and therefore normal cells will potentially be less responsive to the targeted therapeutics given. Perhaps one of the best examples of targeted therapy is the drug tamoxifen. It's actually been around for quite a long time. As you've heard before, there's the female sex hormone estrogen, and estrogen is actually one of the major drivers of breast cancer. And tamoxifen works simply by blocking estrogen binding to the estrogen receptor. So estrogen binds to its receptor, it moves to the nucleus with its receptor, and it initiates a program of gene expression that allows the cell, the cancer cell, to survive, to proliferate, and to invade and metastasize to other organs in the body, which is eventually what kills the patient. Tamoxifen simply works by acting as a competitive antagonist to estrogen. So it prevents the binding of estrogen to the estrogen receptor, and it prevents the cell receiving the survival signals that estrogen would otherwise give to the cell, and the cancer cell dies. Another example of a targeted therapeutic is perhaps one that you've heard about in the media in the last couple of years, given the situation in New Zealand, and that is Herceptin. So Herceptin is a molecule which targets, or an antibody-based therapeutic, which targets a molecule on the cell surface of cancer cells called HER2. So when we target HER2 with Herceptin, we inhibit its action, and again you lose the survival signal for the cancer cell to survive, and the cancer cell dies. We can also indirectly attack a cancer using targeted therapy, and one example of this is the ability of certain drugs to target, I don't think you can see the little dots back there, but to target growth factors that are released from the cancer, which stimulate the ingrowth of blood vessels into the tumour. And obviously if the tumour has limited access to nutrients and oxygen, then its growth is also going to be quite limited. targeted therapeutics also allow us to personalize treatment options for patients. So the example I gave you before about HER2, well, HER2 is only expressed on about 20%, on the tumours in about 20% of the patients. So giving a patient Herceptin when their cancer doesn't express HER2, the only effects that we will see are actually side effects. And so targeted therapeutics allow us to personalize the therapy to a specific tumor in a specific patient, and we can in fact predict the response of the tumor to a specific therapeutic in a specific patient. What we want to do, however, and it's been the focus of the lab for quite a number of years, is to actually identify novel molecules which are acting as drivers for the cancer cell. In other words, allowing them to survive and proliferate and metastasize. And this has been the focus of the laboratory now for more than a decade. We've been reasonably successful. We've identified quite a number of good therapeutics that Joe will talk about, at least one of them. But you actually need quite a detailed knowledge of how a cancer cell functions, which has been a lot of the basis for our work. Because we want to know that the target we select to take to the clinic will actually be efficacious, because going from here to here is at least a few $100 million. Once we've selected a target, there are some really neat and quite smart ways to design new drugs to these novel targets. We then take the drugs that we develop based on these novel targets into preclinical studies to make sure they don't do anything untoward, that they're not incredibly toxic in some unforeseen way. And then once we've gone through all the preclinical studies, we take them into clinical trials to determine if they actually are efficacious in humans. And hopefully if they are, once they come out of clinical trial, they can go into standard use in the clinic. So Joe will now talk to you about one particular target that we've been working on in the lab that is involved in a whole range of functions in a cancer cell. So it's involved in cell survival, cell proliferation, cell migration invasion, and metastasis. And she'll explain to you some of the science behind this particular target that we're working on.
Thank you. Oops. Right, I'm going to tell you about one of the targets we've been working on in the lab, and this is growth hormone. So growth hormone, as its name suggests, is essential for normal growth and development. And so too much growth hormone during development results in gigantism. Too little growth hormone results in restricted growth. And growth hormone, like estrogen, is also involved in sexual maturation during puberty. So the differentiation between male and female sexual secondary characteristics. It's also an essential factor involved in breast development. This is the classical endocrine secretion of growth hormone. So growth hormone is synthesized and secreted in a small gland at the base of the brain called the pituitary. So growth hormone is secreted from the pituitary gland. It then circulates around the blood system and has effects on other tissues. One of the main areas of effects of growth hormone is the liver. So growth hormone actually stimulates the synthesis and secretion of another growth factor called IGF-1. And both IGF-1 and growth hormone will circulate around the body and impact on muscle growth, bone growth, and fat deposition. And that level of growth hormone, or that growth hormone secretion from the pituitary, is not continuous. Growth hormone is actually secreted in a pulsatile fashion. So here we're looking on the x-axis at time over a 24-hour period, and you can see peaks and troughs of growth hormone secretion and quite high peaks and troughs in the young female and the young male. And growth hormone secretion is in response to various stimuli, such as sleep, exercise, and food. Levels of growth hormone will die off or reduce as we get older. So as I said, this is the classical endocrine secretion of growth hormone. Growth hormone is secreted at one site in the body, circulates around the body, and affects a whole range of tissues and organs. But as well as this endocrine system of growth hormone secretion, it's become apparent over recent years that growth hormone is also secreted at other sites around the body. So it has a local secretion and has a local effect on these tissues. Importantly for our work, growth hormone is also secreted in the breast, and as I said before, it's essential for normal breast development. So how are the actions of growth hormone mediated? Well, ultimately, growth hormone regulates gene expression, so it turns genes on and off. But how does it do this? So growth hormone binds to a cell surface receptor. It's a small receptor called the growth hormone receptor. It's found on the surface of most cells. And by binding to the receptor, it triggers a cascade of signals within the cell, the upshot of which is that genes are turned on and off. So growth hormone regulates gene expression. It also has another number of other effects in the cell, but the main effect is regulation of gene expression. And here we're talking about genes which are involved in growth and development. So as Michelle mentioned before, cancer results from an accumulation of changes that occur within a cell which allow it to grow in an uncontrolled manner. Now normally the body has defense mechanisms which would identify these cells and eliminate it. So a number of years ago, Peter's lab up in Singapore asked the question, does growth hormone play a role in cancer? And some of the experiments I'm going to show you shortly have gone towards answering that question. So the first is that growth hormone is produced in cancer, so we can actually see it expressed in tumour tissue. On the left-hand side, you're actually looking at a slice taken through a patient's tumour sample, and the dark staining is the staining of growth hormone in this sample. So you can see that there's localization of growth hormone in the breast tumour sample. On the right-hand side, we're looking at levels of growth hormone protein, and you can see we've measured levels of protein. In the normal tissue, we can detect levels of growth hormone. However, we get increasing levels of growth hormone secreted in proliferative disorders or growth disorders of the breast, including cancer. Now, we also looked at tumour samples taken from 89 patients and demonstrated that growth hormone was expressed in these patients. Now, this is an example of a reverse transcriptase PCR, and we're going to go into how that is done shortly. But for now, you're looking at the top, across the top, different tumour samples. That dark band indicates that growth hormone is expressed by that particular tumour sample. And by looking at this, we found that 60% of the breast tumours that we looked at produce their own growth hormone. We also looked at the growth hormone receptor. And you can see from this that all of those tumours produce that growth hormone receptor band. So all of them express growth hormone receptor, which means that they are all responsive to the effects of growth hormone. But what is the effect? So we've demonstrated here that we've got increased growth hormone in breast cancer. But what's the effect of this growth hormone? So to look at this, we've generated a cell-based model, and we've introduced or forced a cell line to express growth hormone and look at the effects of growth hormone in the cell. And we've done this by transfecting or introducing the growth hormone gene on a plasmid. We're going to go through this whole process. So one of the early advances of cancer research was the ability of scientists to grow cells in culture. And we grow them in the lab today. So we can grow cells in these plastic flasks, and we can look at the changes that have occurred in them which have made them more malignant or more cancerous. So these are the cells we've got in the lab. We've got a large number of human cancer cell lines. On the right-hand side, you're looking at a breast cancer cell line called MCF7. This is one we routinely use in the lab. Just to show you that a little bit more clearly, there's a single cell, there's the nucleus. And the red media that you're looking at in there is actually a growth media. It contains growth factors and nutrients required for cell growth. Now we're hopefully about to show you a small film. Okay, so we're just looking here at a small film. This is one of our PhD students, Graham. Now Graham is removing a cell culture flask from one of our cell culture incubators. He's now taking the cells over to the microscope to check that they're healthy. So that's the view you would see if you look down the microscope at the cells. And that's thousands of cells that you're looking at. He's now taking it over to one of our cell culture hoods. Now this has two purposes. This keeps the cells sterile, so it stops them from growing and getting any contamination from bacteria or fungus. But it also protects the user. Okay. So this breast cancer cell line that I described to you, the MCF7 cells, they don't actually produce their own growth hormone. However, we have induced or forced them to produce growth hormone. And as I said, we've done this by introducing a plasmid vector which contains the entire growth hormone gene. And Marshall is about to take you through this process now.
Thanks, Jo. Okay, this little bit that I'm going to take you through now is going to refer to some of the technologies that you will be covering in your Year 13 course. So some of this will be familiar for you. For some people, others won't have covered it yet. But we're looking at it specifically here. We're not going into detail. We're just looking at it in the context of breast cancer. So Jo wanted 2 cell lines, 2 breast cancer cell lines, one with the human growth hormone gene, one without. So how do they go about getting that gene into a human breast cancer cell? So the first thing they need is a vector. Now a vector, very simply, is something that can transport a gene. Now the vectors that we use most commonly come from bacteria, and they are a circular piece of DNA. And it's very easy to remove them from the bacteria and relatively easy to put them back in again. Now this vector, you'll see it up there on the screen now. No, you won't. There it is. You'll see it's got a little green bit in it. That is the promoter sequence. Now if you've done gene expression already this year, you'll know that in humans and other organisms, the promoter is needed to start the process of expressing the gene. So it's not good enough for us to just put the human growth hormone gene into human cells. It also needs the promoter with it as well. So we've got the human growth hormone gene, we've got the vector, now we've actually got to get the 2 to join together. And to do this uses a rather nifty little molecular technology called a restriction enzyme. Restriction enzymes, the best way to think about them is they're like scissors. They literally cut the plasmid in 2 places, creating a gap that the gene will slot into. So once the gene has been slotted into that, it's obviously got to join with the rest of the DNA, otherwise it's just going to fall out again. So we make use of an enzyme called DNA ligase, and that should be familiar to you because you would have seen it when you studied DNA replication. Ligase joins everything together after the DNA strand has been replicated. And the process of joining it all together is called DNA ligation, and it literally is joining. Plasmid vector, it's got our human growth hormone gene, and it's got our promoter sequence. Now this is just one of them at the moment. We could go through and do this over and over again, but it's actually really fiddly, and it's also not 100% successful every time. So once we've got a successful plasmid, what we can do is take advantage of one of the characteristics of bacteria. Some of you may remember it from year 11, the fact that bacteria multiply very quickly under the right conditions. So we put that vector back into a bacteria, and that process is called transformation. And then we take the bacteria and put it into the right conditions of warmth, food, moisture, all those other things, and lo and behold, after a reasonably quick period of time, we've got quite a large culture of bacteria, all containing that vector, because of course, remember with bacteria, when they replicate, the daughter cells are identical, asexual reproduction. Once we've got that culture, the next thing we do is purify it, so we end up just with those vectors. So now we've got lots and lots of the vector, all containing the human growth hormone gene. So the next job is to get it into the breast cancer cell. Now, that's not quite as easy as it sounds, and those of you that have done the biotechnology will know that. There are lots of different techniques for getting a vector to insert its DNA into the nucleus of a new cell. None of them are highly successful. One you may have covered is the use of electrical current, and that's the technique that Peter and Jo's group use here. So an electrical current is applied to the membrane of the cell, and by some mechanism, which I don't think anybody quite fully understands, the vector then moves into the nucleus, and the gene that it's carrying is therefore available to be transcribed and produce the human growth hormone. Now, any good experiment needs a control. Because if you're going to go and look at what effect is this human growth hormone having on the breast cancer cells, you need to be sure that any differences you see are due to that human growth hormone being expressed, not due to the fact that you've been applying electric shocks to breast cancer cells and growing them in funny conditions in the lab. So what they do is they set up a control line of cells. So it's treated exactly the same way, including having a vector added into it. The only difference is that vector doesn't have the growth hormone gene in it. So what we end up with is those two cell model lines. One is going to be expressing human growth hormone, the other one is not. And Joe's now going to come and tell you what they found out, what sort of differences they saw between those two cell lines.
Thank you. Okay, the first thing we noticed, so we've now got a cell-based model where we've got two cell lines, one which doesn't express growth hormone and the other which does produce growth hormone. So the first thing you immediately notice when you look at these cell lines is that they look different. They look different when we look at them down a microscope. On the left-hand side, the control cell line, the cells are closer together. They have more cell-to-cell contact and they tend to clump. Whereas on the right-hand side, the cells are more spaced out, they have less cell-to-cell contact, and they're more likely to migrate across the tissue culture dish. They've actually got a more aggressive phenotype. Now, when we look at the growth of these cells, so as Michelle mentioned earlier on, there are some hallmarks of cancer, and the first being uncontrolled cell growth. And when we look at the growth characteristics of these cells in culture, we see that the growth hormone cells, producing cells, grow faster. So this is an experiment where we've counted the number of cells in culture over a period of time. And you can see that the growth hormone cells grow much faster than the control cell line. We've also looked at a type of apoptotic cell death. So as I mentioned, the cells producing growth hormone grow faster. And this is similar to cancer in that cells grow in an uncontrolled manner, which would normally be picked up by cell defense mechanisms and eliminated. However, as well as growing faster, the growth hormone cells survive better as well. So we've looked here at a type of cell death called apoptosis, and you can see in the growth hormone cells, or the cells that are producing growth hormone, that we've got a lot less apoptotic cell death than in the control cell line. As cancer progresses, cells acquire the ability to migrate and invade surrounding tissues. We've also demonstrated that growth hormone promotes cell migration and cell invasion. Another hallmark of cancer is angiogenesis, and tumours or cancer cells acquire the ability to attract blood vessel growth into the tumour, and this allows the supply of oxygen and nutrients to the tumour, and it facilitates its growth. In recent studies, we actually demonstrated that growth hormone produces or promotes tumour growth and promotes growth of blood vessels into the tumours. And that's actually been quite a recent study in the last couple of years. Another hallmark of cancer is immortalization. Cancer cells, or normal cells I should say, normally have a limited lifespan. So they have a limited number of cell divisions. However, growth hormone promotes the activity of an enzyme called telomerase. And telomerase adds telomeres onto the ends of chromosome, thereby extending the replicative capacity of the cell. So it extends the lifespan of the cell. So basically, to sum that up, and this actually summarizes about 12 years of work and a whole lot of publications in a single slide, when we introduce growth hormones into these cells, they grow faster, they survive better. They're more likely to migrate and invade the surrounding tissues. They can form tumours and then promote the growth of blood vessels into those tumours. And it looks like growth hormone actually contributes to all these stages of growth hormone. So abnormal secretion or deregulation of growth hormone in cancer promotes cancer progression. In recent studies, we're actually now beginning to look at some chemotherapeutic drugs and show that the cells which express growth hormone are actually less sensitive to some of the drugs which are used to treat cancer. And this is actually quite important because development of resistance to some of the chemotherapeutic drugs is actually an important or a difficult hurdle to overcome in cancer therapy, and it can decrease the prognosis for patients. So So of course we're actually interested in looking at ways of inhibiting growth hormone action. So we're developing strategies to block the actions of this hormone to use as a therapeutic treatment in cancer. And we hope by blocking growth hormone action in conjunction with some of the conventional chemotherapies that we might be able to improve the prognosis of breast cancer patients. Now, we just want to move on to something else. We've shown that growth hormone has an effect in cancer, but how does it have an effect in cancer? What is the mechanism by which growth hormone promotes cancer progression? So we were actually interested in looking at some of the genes that were regulated by growth hormone, because these genes themselves might also be important in cancer. And Michelle is now going to take you through some of the processes or the techniques we use to look at gene expression.
Okay, so back to biotechnology again. So for some of you it's going to be new, for others it's going to be going over stuff you've already done in class. So as Joe said, they're really interested in finding out what exactly is this human growth hormone doing, and they think what it's doing is affecting the regulation of genes in the cell. So they needed to be able to measure expression of other genes inside those cancer cells. So how did they go about doing this? Well, you might be a little bit surprised that they do it by measuring the messenger RNA. Now, I wonder if any of you can make the connection between gene expression and messenger RNA. You all should be able to, because we talked about it in the last seminar. Remember this? Remember genes, if they're going to be expressed, they produce messenger RNA, then messenger RNA is turned into a protein. So if we look at the levels of messenger RNA in a cell, we're actually going to get a pretty good idea of how active that gene is. Is it being switched on? Is it being switched off? Is it being expressed at higher levels than normal? Is it being expressed at lower levels than normal? And that's what Joe and Peter's group have actually been doing. So how do they go about doing it? How do you actually go about measuring the levels of messenger RNA in the cell? Well, the first thing, there are 3 techniques that can be used. 2 of them involve PCR. For those of you who haven't done biotech yet, the rest of you will know this, PCR stands for polymerase chain reaction. And very simply, it's a technique that amplifies DNA. And it can be used to, as I'll show you shortly, to work out what the levels of messenger RNA are doing. However, both of these 2 forms of PCR have one disadvantage in that they only look at one gene at a time. Now, when you consider we've got tens of thousands of genes in the human genome, and they're all doing different functions, it can be rather slow if you're going to go through them all one at a time. Of course, they rule out the obvious genes that have got nothing to do with cell growth, but even so, it's a rather slow process. So in recent years, a new technology's come in, which is called microarrays, and they have the ability to analyse literally thousands of genes at one time. So let's look at each of these as we go. So the first one, reverse transcriptase PCR. Now, PCR amplifies DNA. So somehow, from the messenger RNA that the cell is producing, we've got to get some DNA that can be amplified by PCR so that we can actually see it, to see what's going on. Now, the process of doing that is called reverse transcription. Now, if you haven't met it before, look very carefully at that name. Reverse, you'll know what it means, go backwards. Transcription, part of protein synthesis. So maybe it's got something to do with protein synthesis. Remember this from the last seminar? Normally transcription runs from DNA to messenger RNA. Reverse transcription is literally the reverse. The process takes those codes on the messenger RNA that have been produced by the gene we're looking at and uses it to make a piece of DNA that is called complementary DNA, or cDNA for short. So we've got our cDNA, that's then amplified by PCR using an enzyme called DNA polymerase, which is the one that you've met before in DNA replication. Now, once we've got an amplified amount of the cDNA, we can then put it into a gel, run it through gel electrophoresis to see whether or not the messenger RNA was present in our original cells. Now, at this point, Jo and I are going to do a bit of a team approach here. She's actually going to show you some of the results of those of this reverse transcription PCR.
So hopefully we've got a short clip very shortly to show you the process of RT-PCR. This is a reverse transcriptase PCR. So you're actually looking at a postdoc, Michael, from our group. He's setting up individual reactions for RT-PCR. So each reaction will be looking at gene expression of a single gene. This has been loaded into a PCR machine, and this machine will allow amplification of the cDNA target. That probably takes a couple of hours to run. And when it's finished, we can load them onto a gel and conduct electrophoresis or gel electrophoresis. So here he's loading each of the reactions, so the amplified product, onto a gel. An electrical current will pass through the gel and separate the bands out on the basis of size. So we can then look at the amplified band. Okay, and here's what one looks like. So you've already seen this RT-PCR experiment. This is where we used RT-PCR to look at the levels of growth hormone in the tumour samples. So again, tumour samples across the top, and that dark band is actually the amplified growth hormone gene following an RT-PCR experiment. Now, along the bottom here, we have what we call a loading control. This is actually a housekeeping gene, or what we call a housekeeping gene. A housekeeping gene is a gene which is expressed supposedly at the same level in every single cell. So it should be the same in every sample. And we actually use that to make sure that we didn't stuff up when we were setting up. So we haven't loaded the wrong amount of cDNA or unequal amounts of cDNA. So this is what we should see. It should look pretty equal across the bottom. And that indicates that we added the same amount of cDNA to each tube and the same amount of the reaction onto the gel. Here's another example. Here we've used reverse transcriptase PCR to validate our cell model. So you remember we've transfected our cell, MCF7 cell line, with an expression vector or plasmid containing the growth hormone gene. And after we've generated these cell lines, we need to check them and make sure that they are expressing growth hormone. So we've conducted RT-PCR to look at expression of the growth hormone gene. You can see on the left-hand side the control cells don't express growth hormone. On the right, we get a band indicating that we've successfully transfected these cells. They're now producing growth hormone. And again, underneath is a loading control to make sure that we had equal amounts of cDNA in each reaction. Here's another example. Here we're looking to see whether growth hormone regulates genes involved in growth and development. And on the right-hand side, these three genes, c-MYC, cyclin D1, and BCL2, are all involved in growth and survival. If you compare the control cell line on the left-hand side to the growth hormone producing cells on the right-hand side, the band's brighter, it's more intense. And this indicates that growth hormone has increased the expression of these genes. So it's upregulating gene expression of these genes involved in growth and survival. Underneath again, we have the loading control, so showing that we've got equal amount of cDNA. I will now pass back to Michelle, who will tell us about real time.
Right. Reverse transcription PCR has one disadvantage. It doesn't actually allow you to quantify the amount of messenger RNA that was there to start with. All it does is tell us that that gene was being expressed or not being expressed. So the next version of PCR is called real-time PCR, and that actually does have the ability to quantify. In other words, to give us an idea of how much messenger RNA is being produced by those genes. So it starts off exactly the same as the normal PCR, except when the PCR is run, there's a fluorescent dye involved. And that is actually able to be analysed, and we can quantify the level of the fluorescence. So more fluorescence means there was more of the cDNA product, which directly relates back to the amount of messenger RNA there was at the start. So this means that the scientists can actually say, not only is this gene being expressed in the presence of human growth hormone, it's actually being expressed at a higher level than it would normally have been. But as I've said and Joe said, we can only do one gene at a time with this. So that means it's a very, very slow process because there are so many genes that are potentially involved in it. And that's where microarrays come into it. Now, microarrays allow the high throughput analysis of tens of thousands of genes. And to do that requires an array. And Joe's just handing me one now. DNA chip or a gene chip. In my hand, I'm holding something that has the entire human genome on it, all 24,000 thereabouts genes, plus a little bit more, as Joe will tell you shortly. So we can actually, with this one thing, we can actually analyse the expression of all the genes in the human genome. So it's a pretty major impact on our ability to look at gene expression. Now, how this is used, I'm not actually going to cover in the seminar today, but there is a diagram in the seminar paper which takes you through it. But basically, microarrays have lots and lots of uses. In the use that the ligands scientists have been using it for, they have been using it to compare those two cell lines that we've been talking about all the way through. The cell line that's producing human growth hormone and the cell line that's not. Now, at the end of when you've used a microarray, what you end up with, whoops-a-daisy, don't know what I did there. What you end up with is a picture that looks like this, and these coloured dots actually mean something. The yellow dots means that the level of the gene expression in the two samples is the same, so there's no difference between them. A red dot means that in one of the samples, the level of gene expression is higher than it is in the other one. And a green dot means it's lower. So looking at the colours can actually tell the scientists an awful lot about what's going on with the expression of the genes in these two samples. And this is where Jo comes back in again to show you what she's done.
Just like to point out that, okay, there are 24,000 genes on this slide, but each gene has 26, or an average of 26-fold coverage. So there's 26 roundabout probes for each gene. So it's actually about 700 or 800,000 spots on this slide, which can be individually characterised. So it's actually quite a few. So we wanted to look at the expression of genes, as I mentioned before. Earlier on I showed you RT-PCR, which looked at 1 or 2 genes at a time, and that's time consuming. So if we wanted to look at every gene in the genome, obviously that would take us a very long time. So we conducted microarrays, and we looked at the control sample compared to the cells which were producing growth hormone to see which genes growth hormone had turned up and down. This was done a while ago, so we had an older chip, or an older version of that gene chip. So there were only 19,000 genes on our one. So we analysed 19,000 genes. We found that 305 were actually regulated or exclusively regulated by autocrine growth hormone. Now, a number of these genes had already been described in cancer, so they already had a role in cancer, whereas quite a few of them did not, or they did not have a role previously described in cancer. We are currently investigating some of these as potential novel therapeutic targets, and we'll take them through the same processes that we have done for growth hormone and look at their effects on cancer cells. All right, thank you.
Okay, so that's the end of the seminar. You all know the routine from here. We'll do a question and answer session very shortly. And when you go back to your schools or go into your post-seminar workshops, you are, of course, going to be looking at the post-seminar challenge questions. Now, I'm not going to actually talk you through the questions today because I've heard that there's been an incredibly busy discussion on the seminar chat and lots of questions coming through from there. But just a reminder, where you get the benefit from these seminars, it's not just being here today or being in our remote audience and listening to it, it's going back, looking at those challenge questions and having a go at answering them. And if you have a go at answering them, either posting your answer on our wiki or at the very least giving them to your teacher so your teacher can give you feedback. It's the practice in answering these style questions that will give you the best practice for your exams at the end of the year. Okay, so we've already had a question come through on our question page, and this one's from Dilworth, is it? There's a bit of applause in the audience in front of me. The student didn't put a school. Can polymerase be artificially produced and somehow used to prolong life? Telomerase. Okay, there's been a typo when we transferred it. So.
Okay, telomerase, of course, in cells does prolong the lifespan of the cell, but as you've seen from some of the talk today, telomerase also is a key factor involved in cancer progression. So a large range of cancers actually have increased telomerase. So you'd be worried about using telomerase as an anti-aging therapy for its effect in cancer or potential to promote cancer.
Okay, thank you. Right, do we have any questions from the live audience? Not today, maybe. Okay, Anna, I'll throw it open to you for some of the questions that have come through from the seminar chat. Hello, Anna. We can see a question, we can't hear Anna. Okay, I'll read the question. Could cancer form if a mutation occurred only in the intron of a gene, taking into account that cancer forms from accumulation of mutations? And that's Bridget from Christchurch Girls High School.
Actually, it can. I mean, often we talk about mutations in exons or the coding regions of genes, which might confer a specific growth advantage or change in amino acid, which then might result in a mutation in the protein. And this has the potential to cause cancer. But, okay, so we're getting into something here called the junk DNA. There are a lot of regulatory regions in between those coding regions of genes. And actually, there's only a smaller part of our genome is made up of coding regions. And a large amount of it is just repetitive DNA. So for quite some time, scientists thought that this was just junk and wasn't worth anything at all. But it's become apparent there are a lot of transcribed regions from so-called junk DNA, and including transcribed regions which might not go on to form proteins, which come from introns and from those large repetitive regions. And mutations in those have been shown to contribute to cancer, so yes, they can do.
Okay. All right, we've actually got 10 questions coming out of the seminar chat today, which is really good. So people in the live audience here, if you do have a question, if you just stand up at some point, I'll see you, and then I'll come to you once the question's been answered. Now, we're also having a few problems, so you're not going to hear Anna's lovely voice today, so we're just going to have to go with the question on the screen. So, are there side effects from taking tamoxifen, particularly in sexual development, as it prevents estrogen from binding? Another one from Christchurch Girls High School.
I guess you wouldn't expect teenage girls to get breast cancer, so you wouldn't actually be treating teenage girls with tamoxifen during their period of sexual development. So tamoxifen is usually a treatment that's given to women, either postmenopausal or premenopausal, when their sexual development is actually finished. So no, that wouldn't be any worry in terms of a clinical application.
Great, thank you. Okay, next question please from the seminar chat, or Anna's voice if they managed to get it working. Question? I know there are a lot. Here we are. That's the same question. It's on its way. We're having all sorts of technical problems today. And of course everything worked perfectly at the beginning when we tested it. So anybody in the audience got a question that we can... Yep, wonderful. We have an audience question.
I wonder, are there more than one cancer therapies carried out on one patient?
Okay. Right. If anybody else from the audience can actually go to the mic, but I'll repeat the question for those of you that didn't get it, as our remote audience would have. Are there more than one cancer therapies is there more than one cancer therapy carried out on a single patient?
Nowadays, most clinicians understand that so-called combinatorial treatment, where you combine different chemotherapeutics or different agents to treat one particular cancer, is actually more effective than a single agent alone, or using a single agent sequentially. So it is preferable actually to be able to design a chemotherapeutic regime or cocktail that you can give to a particular patient based on what type of particular cancer they have to achieve maximum effect.
Great, thank you for that, and thank you, well done to somebody in the live audience. We haven't had anybody for a while. Okay, next question. As our immune system does not recognise cancer cells, is it possible to develop a vaccine or therapy that would allow our immune system to recognise and target them?
Immune therapy of cancer is quite complex. I mean, I'm sure you're all aware of the cervical cancer vaccine, but that's actually a vaccine against a virus, so it's preventing infection with a virus. So that's why it's effective, because it's just like an influenza vaccine. You can get vaccinated and prevent your infection with a particular virus. In terms of breast cancer, there's not a whole lot of evidence that breast cancer is caused by a virus, although some people are actually looking at ways of potentially activating the immune system to destroy a cancer cell, a breast cancer cell. These experiments have been going on for quite a number of years now. In some instances, I'm aware of a company in Sweden, for example, in terms of colon cancer. They have developed a way to sort of stimulate the body's own immune system. They take out your immune cells, culture them up, do some tricks and put them back, and it's having quite some success. They, however, haven't published what they're doing. It's a company's secret. So in terms of breast cancer, probably at this stage a limited application.
Thank you. Okay, we've got another question from the live audience. Hold on just a moment. Try again. The mic wasn't on. Okay. Say it again and I'll repeat it.
Is it possible to isolate the cancer tumours from the blood supply?
Okay. Is it possible to isolate the cancer tumours from their blood supply?
It's possible to identify cancer cells in the blood circulation once the tumour has metastasized. And these cells are called disseminated tumour cells, or DTCs. That's possible. You can actually then take these cells out of the circulation and culture them, but in terms of a therapeutic treatment, it probably wouldn't be much use, except in terms of determining whether the patient has metastatic disease, which would then affect the prognosis. Obviously, if a patient has metastatic disease, they have a worse prognosis. So yes, you can detect cancer cells in the bloodstream of patients with metastatic disease. And you can actually do this by using a series of gene markers which are specific for example, mammary carcinoma cells. And we're actually working on some of these at the moment.
I think that was asking whether you could isolate the tumour from the blood supply as well.
Oh, did I misinterpret the question? Can you ligate the artery feeding the tumour? Usually when tumours form, they're actually fed from multiple different angles in terms of blood vessels. So you have a whole lot of very small capillaries feeding the tumour rather than one large blood vessel. There are, however, a number of so-called anti-angiogenic therapies. One of the most potent angiogenic molecules is a molecule called VEGF, vascular endothelial growth factor. And there is a drug, an antibody-based drug against VEGF, which can inhibit angiogenesis in tumours. The success for this drug, called Avastin, so far, I think has been limited to lung and colon, with limited success in breast carcinoma.
Great, thank you, Peter. Okay, back to the wiki. We've got a lot of questions coming through there. All right. Why do Western countries have a higher incidence of breast cancer, Joe?
Okay. That's a very good question, and like I said, we really don't know the answer to that. It's suggested that it could be due to dietary factors, so we're also, Western diet is higher in fat, and that has been associated with a higher breast cancer incidence. There are also social or cultural factors. For instance, Western women are beginning to have less children and having them later on in life. And some of the Asian countries, there are more children in the families. So really it's ongoing investigation, and it's actually really interesting because if we can identify some of these factors which increase the incidence of breast cancer in Western countries as opposed to Asian countries, it means that we can potentially identify some of the risk factors that we can modify and potentially reduce the incidence of breast cancer. It's actually something I've become quite interested in after working here at the Liggins, because I'm beginning to link in with people like Dr. Sapoda last week, and we're becoming interested in the effects of maternal nutrition, so diet during nutrition during pregnancy, and how that might affect cancer risk later on in life. But yes, interesting question.
Okay, we're on a roll tonight in the live audience. We've got another question. Would you like to come forward to the mic, please? Yep, we've actually now got it working, so.
You sure? Oh, I've heard about nanotechnology being used to like try develop a way that something can go target and kill the cancer or something, but I was just wondering about if New Zealand does any research on that and how far are they along in the research in nanotechnology, that kind of thing.
Good question. You've got the two scientists looking at each other.
Well, I'm aware of some people working with nanotechnology. I'm not sure whether they're doing anything towards cancer research, but you know what? I'll look it up for you and I'll post an answer on the wiki site. How's that?
Brilliant. Thank you. Okay, another question from the wiki, from the seminar chat. If someone has cancer, can this then be passed on to your offspring? I think I can probably answer this one. I'll start it. I want you, before you have a think about that question and have a think about what type of cells is the cancer found in. Body cells. What cells produce the sperm and the egg? Gametes, yes. So where does the cancer need to occur, or the change need to occur if it's going to be passed on? Right. So no, a very simple answer is, and I'll wait for the scientists to add the complicating factor, no, the simple answer is no, cancer in a body cell would generally not be passed on. Joel, Peter, do you want to add anything?
Yeah, I'll just add in there as well that there, in most types of cancer, there are specific cases of hereditary cancer. So for instance, in breast cancer, mutations in genes such as BRCA1 and BRCA2 are associated with a significant increase in breast cancer risk. So yes, that is hereditary. It is passed on to offspring, and it also results in an ovarian cancer and can contribute to prostate cancer as well. So there is a genetic component often with cancer. Again, as in with breast cancer, most of the cases are sporadic, so there's no known genetic factor associated, and that can be passed on. But that can be passed on then, yeah.
But in that case, what's actually been passed on is the risk factor, in this case the mutation, not the cancer itself. Okay, next question. If someone has bigger breasts, does this make them more susceptible to breast cancer? How does this relate to density?
I mean, if you have bigger breasts, you have more breast tissue, so to speak. So basically, your chance of a breast cancer arising in more breast tissue are greater. Although, Joe, correct me if I'm wrong, I don't think there's really any studies showing any tight correlations between breast size and tumour incidence. As Joe said, there is the correlation between height and tumour incidence.
It seems to be the density, not the size of the breast. So it's how dense the breast is, the epithelial and stromal components of the breast.
But the hormones that make you grow tall are also the hormones that contribute to your breast density. So that's why you get the association.
Okay, that was the last of our questions from the wiki, but I believe we've got one more from the live audience. This is a record. Well done. Would you like to come forward to the mic, please?
I might be completely wrong here, but I have heard possibly that cancer therapies often have high success rates in pediatric oncology, so the treatment of younger individuals. I was wondering if that's true and if you might have any suggestions why that might be.
That's true in terms of childhood leukemia and other blood cancers, so to speak. And it's probably true because the changes that occur to give children these particular types of cancers are very well defined. So we know exactly what's happening in the cancer, and drug companies have put a lot of money into really developing treatments against that defined problem. So you can actually, in a lot of instances in childhood cancers, get cures. There are a lot of other childhood cancers, though, that are incurable. So I think probably what you're hearing is the blood cancer story. Yeah. When you're an adult and you get cancer, you're probably likely to get many different variations in the type of cancer. So you can do gene profiles on a whole series of breast cancer, for example. And while you can pick out commonalities, the breast cancers are actually quite different between each tumours. I'm sure you've probably been aware of the recent sequencing data that's come out of the cancer genome. And each individual cancer can have up to 30,000 different mutations. So we're talking about a very heterogeneous type of cancer in adulthood, as opposed to the very defined mutations or linkages or chromosomal aberrations that we get in some childhood cancer.
Thank you. Right. Oh gosh, there's another one. Quickly, we've just got time for the last question from the floor.
Does the incidence of breast cancer increase when the woman is on progesterone or estrogen-based contraceptives?
There is a slight increase in risk associated with contraceptive pill. There are several complicating factors there, but in general we say there is a very slight increased risk with contraceptive pill. Yep.
In terms of breast cancer, but actually in terms of endometrial or ovarian cancer, you actually decrease your risk. So, depends.
Great. And on that very complicated note, like a lot of things in biology, that brings us to the end of our seminar. Well done, everybody. That was a really amazing collection of questions. Remember that there will be a prize awarded for the best question from the seminar. And my apologies to those of you who got prizes in the seminars last term. We haven't sent them out yet, so don't panic. We had a bit of a delivery problem with them. They finally arrived today. So thank you very much. I'm really looking forward to seeing all your responses on the wiki. And remember, if you've got more questions, go into the wiki and post them, and Deb and Peter will, and I will do our best to answer them. So thank you and good night.